Okay, good afternoon. So I think last time the recording actually worked quite well. So it's already on YouTube online. So I'm going to try to keep doing that. No guarantees though. I think the only downside is that when I'm recording on screen capture, when I was in Hong Kong, I was just talking to the computer, so you heard me much better. Whereas now I'm sometimes here and sometimes there, but I feel like that's probably a good trade-off. So thank you for submitting your homework assignments. I've heard from all but two people. So once I get all the submissions, I'll go over them in detail with you. Obviously, everybody's submission is going to be different, but from my looking at your submissions so far, it looks really good, right? It looks like you guys really got the point and got the idea. But the point of the assignment is really just to get you guys to look into brain space, just to get a little bit more hands-on experience into scrolling through this three-dimensional voxel space that is the brain. Now today we're going to finish talking about audition. Specifically, we'll talk about a couple of topics in music and the brain that I've worked on and find very interesting. And then we'll move on to chemical and mechanical sensations. All right, so we finished talking about kind of general subcortical processing of auditory stimuli the last time, and we've reached the topic of higher auditory functions, right? So that includes speech and music, so everything that is at the level of the primary auditory cortex and beyond. But today we're going to talk about a couple of special populations that have an unusual behavior, especially in music and the brain. And I'd like to talk a little bit about people who are especially good at something and people who are especially bad at something. And that something is music. So absolute pitch. Does anyone hear perfect pitch? Absolute pitch? Okay, besides myself? Okay. So this is the ability to categorize pitches, musical pitches, without a reference. And so that's so most periodic sounds, as we talked about the last day when we were talking about sound stimulus, they're characterized by systematic variations or changes in air pressure in the way they hit the eardrums, right? And so the way these sounds hit your eardrum, if they recur in a very periodic, predictable manner, that gives you the sensation of pitch. Right, so all the music that you hear has certain pitch. It can be high pitch or low pitch, and that's really kind of correlated with the fundamental frequency at which the air molecules are vibrating. Right, and so but what people with absolute pitch tend to do is when they hear a certain periodic vibration, they'll say, "Oh, that's an A," right, and that's related to the way in Western music we've come to categorize sounds into different pitches, and that's a pretty rare characteristic. It's less than 1% of the general population, but it's very unevenly distributed across different parts of the population. So there are some genetic contributions. So if your mother or some family member of yours has absolute pitch, you're much more likely to also have it. So there's some genetic contributions. People of East Asian descent are much more likely to have it, and that's also been related to genetic contributions, but also to a relationship with tone language. So we know that people with East Asian descent more likely to speak Chinese or Vietnamese or some language that conveys meaning using different pitches, right? And we'll talk much more about this when we talk about language, but these tone languages seem to be correlated with the characteristic of absolute pitch. Then there's also developmental contributions. Right? So people with musical training are much more likely to have absolute pitch, and the kind of absolute pitch you might have actually depends on the kind of musical training you have. So there are some people who, for instance, who have violin musical training, violinists tend to have absolute pitch for higher regions of the keyboard, so higher notes. Cellists tend to have absolute pitch for lower regions, so lower pitches, and then pianists tend to have absolute pitch for just the white keys of the keyboard and not the black keys of the keyboard. So just to give you an idea of how we can test for absolute pitch, we have some certain online tests that we develop for these things, but it's really quite a simple, this is just an example of what the interface looks like, right? So you click, you hear a tone, and you have to categorize it, right? So just to give you an example, this is just a demo, right? So you'd hear a tone, right? So that's an A, right? You would say that's an A, and that's a D, and that's a B, and that's an E, and that's an E, and that is a C sharp, right? So most people would be randomly guessing, but if you have absolute pitch, when you hear it, you would say, oh, that's definitely an B sharp or whatever. So how do we quantify this kind of behavior? So we can quantify this as a function of your deviation in your response as a function of semitones away from the correct answer. Right? So a semitone is just one step in the Western musical scale. So if your answer is 0 deviations away, 0 semitones deviating from the correct answer, then that is the correct answer. And so most people without absolute pitch are just randomly guessing. But then, in contrast, people with very accurate absolute pitch, we'll call those AP1 possessors, they're almost always correct. Right? So, and when they're wrong, they're just one semitone away from the correct answer. And then there's some people who are kind of in between, right? We'll call those AP2. So those people are still way, way, way above chance, right? They're still much, much more accurate than the non-absolute pitch possessors, which is most people in the population. But they're not quite as accurate as the AP1 group. And so these two groups of people, AP1 and AP2, we generally lump them together to say that they have absolute pitch abilities. But in general, we like to think of them as having an enhanced perceptual categorization abilities, right? So some of you might be interested in, for instance, other orders like autism, right? That is also characterized by enhanced perceptual categorization. Right? So people on the autism spectrum are much more likely to say, kind of over-categorize certain things. Right? So, kind of unusual items, I guess everyday items like chalk, for instance, they might say, oh, that's a long piece of chalk, that's a particularly short piece of chalk. So they might categorize things in ways that people not on the spectrum might not. Right? So there's this enhanced ability to perceptually categorize things. And that's also related to some individual differences in structure and function of the auditory cortex. So this is a nice classic paper from the 90s. Dr. Schlau and colleagues looked at T1 images. So these are MRI structural images of the brain of people with absolute pitch and people with no absolute pitch who are still nevertheless have high musical training, and then people who are non-musicians, right? So no musical training. And what they found was this striking asymmetry in people with absolute pitch, asymmetry between their left and right planum temporale, right? So in the last class, we talked about the auditory cortex, right? And I said there's a bunch of different words that people use for the anatomy of the auditory cortex. That includes Heschel's gyrus, superior temporal gyrus, planum temporale. Planum temporale generally refers to a part of the superior temporal gyrus, right? So this here is a picture of the schematic of the whole brain, right? And so, of course, the auditory cortex, as we talked about, is right underneath the Sylvian fissure, right? So the Sylvian fissure is this fold that's right in between the temporal lobe and the frontal lobe and the parietal lobe, right? So that's right there. And right underneath that Sylvian fissure, we have the primary auditory cortex, and that's part of the planum temporale. And what Schlau et al. Saw is that people with absolute pitch tend to have a much larger planum temporale on the left hemisphere relative to the right hemisphere. So while the right hemisphere planum is about the same volume as everybody else, the left planum is larger in people with absolute pitch. And so some years later, I was working in Gottfried Schlau's lab, and we further did a diffusion tensor imaging study looking at a similar population of people with absolute pitch. And specifically what we did was we did diffusion tensor imaging of people with in the AP1 group, right? So people who are very, very accurate, accurately able to categorize pitches. And by the way, these people in the AP1 group, they're so accurate that it's a little bit compulsive, right? They might hear sounds in the wind, and they'll say, oh, that's a C sharp, right? So there's a sort of hyper-categorization ability. And then in contrast, there's an AP2 group, and then there's the non-AP group, which is most people, right? And these here are tractography images that are overlaid on a fractional anisotropy images. So that's just DTI images, and then so that's the black and white part. And these are coronal slices, right? So we're looking at brains that are looking into the screen here. And then in color, I'm showing the tracks that we were able to find that connect the superior temporal gyrus, right, which again corresponds to roughly the same part of the brain as the planum temporale, which I talked about just now, and its connections to the middle temporal gyrus. So I think the main point is that we're trying to identify white matter pathways that are in the auditory perception and categorization areas, right? White matter pathways that link the perception and categorization areas in the brain. And what we can see here, I think, is that the absolute pitch 1 group, right, so these are the super hyper accurate categorizers, have much more track volume, especially in the left hemisphere. So left is left on this picture. And so the AP1 group possesses much larger white matter pathways. And so this provides a little bit of support for this idea of hyperconnectivity, right? So these people that tend to over-categorize things or be hyperactive in their ability to categorize sounds, that might be subserved by an abundance of connectivity structurally in the brain. So I think that's pretty interesting. And it remains to be seen, right, what other kinds of differences these people might also have. Now, so that's one end of the spectrum. So some people who are exceptionally prone or have a strong tendency to hear more accurately or in a more hyperactive manner, so hyper sensitivity to sound. And then in contrast, we could also think about people who are not, who are particularly insensitive to sound. I actually feel really bad using Hillary Clinton as an example after the events of the last couple of years. But this is actually an example I'm about to show you. I think I have it. It's an audio file of Hillary Clinton singing the national anthem in 2007. This is before all the recent events. It's kind of, she doesn't realize that the microphone is on her, and this is the last line. Okay. I mean, this is like one of my heroes, right? But and like obviously a very good speaker and very good with her choice of words and her language and like very hyper intelligent person. But somehow like it doesn't seem to, the musical ability doesn't seem to match up with the other abilities. So just to give you more of an example of what tone deafness or congenital amusia means. I'm about to play you another example of an inability to sing in tune, and this is the singer is singing the Happy Birthday song to me. This is one of my previous research subjects who came into my lab, but she at the time was a med student at Harvard Medical School, so a very highly accomplished student, who had also had 8 years of violin training and was a native Chinese speaker. So she's got some tonal language experience, and she's got a lot of musical experience, and she's a good student. So this is what it sounds like.
Happy birthday to you. Happy birthday to you. Happy birthday, dear Psyche. Happy birthday to you.
Anyone think this sounds good or normal? Okay, so if you think it sounds normal, come talk to me afterwards. All right, so this is an example, a prototypical example of congenital amusia. Amusia is the lack of musical ability, right? So throughout the course, you'll hear about whatever, yeah. So amusia is lack of musical ability, agraphia, a lack of writing ability, dyscalculia, a lack of mathematical ability, and so on. But today we want to talk a little bit about amusia, and there's 2 types of amusia. You can have acquired amusia, which is when you lose musical ability as the result of a brain injury, right? So there are some people who, due to stroke or tumor or some neural insult, end up with a lesion somewhere in their brains, and that's impinging on areas of the brain that you need to have normal musical ability, and that's acquired amusia. In contrast, this is a case of congenital amusia. So in other words, you're born with it, right? So, or you never knew, or you don't know of one brain incident that led to your loss of musical ability. And so it's characterized by inability to sing in tune. I'm sure a lot of you might know somebody who's just spectacularly bad at singing. But there's a slight difference between spectacularly bad at singing, where they know that they're bad at singing, and spectacularly bad at singing where they're not even aware of how or what is unusual about their singing. Right? So we're looking at the latter category here. And the incidence, it depends on who you ask. So 1 out of 7 people think that they're horrendously bad at music and will therefore self-identify as being tone deaf or congenitally amusic. Most of those people are actually not. Right? So there's various reasons why you might not be a particularly good singer. But the real incidence is probably around the 4% mark. But that 4% mark is identified using a neuropsychological battery of tests using, it's called the MBEA, which stands for the Montreal Battery for Evaluation of Amusia. And how that works, you can just Google it and you can take it if you're interested. It's just pairs of melodies, one after another. They're kind of composed for the experiment. They don't sound very good in my opinion, but you can, you just have to listen to these pairs of melodies and you just have to say whether they're same or different from each other. And if you score below a certain cutoff point, then you get categorized as being amusic. So there's some difficulties with these kinds of categorizations that are true of all kinds of standardized tests. But in our lab, we also use a more kind of psychophysical or kind of bottom-up hearing test to identify people who might be congenitally amusic. And that's using a pitch discrimination threshold. And so feel free to go onto this website and take it yourself. But it's just these pairs of tones, one after another. And you just have to say whether the second tone is higher or lower than the first. And if you score below a certain cutoff, then you get categorized as being tone deaf or amusic. So a number of people have looked into what makes people's brains amusic. What makes people amusic? What is different about the brains of people with congenital amusia? Now, with acquired amusia, it's kind of obvious, right? So if you get a lesion and at the same time you lose your musical ability, you can start to relate that lesion to that musical ability. And one interesting kind of follow-up to that is whether you can regain some of that musical ability. And there's some work just coming out in this couple of years that shows that the extent to which certain parts of the brain recuperate their abilities or their functions and their structures are correlated with your recoverability of musical functions. But this is just looking at congenital amusics. And what they found, looking at cortical thickness measures, is that sometimes less is more. So what was curious about their finding is that there was an inverse relationship, or a negative correlation, between musical score and cortical thickness. So in other words, the people who were worse at music, at the musical tests that they would use, which is the Montreal Battery for Evaluation of Amusia, those people tended to have thicker cortical thickness in a couple of regions, specifically the right superior temporal gyrus, which is one of the kind of seats or important areas for music processing and other kinds of auditory processing. And the other area is the inferior frontal gyrus. And so people who were worse at these tests tended to have thicker gray matter in these regions. Now, so that previous paper identified a couple areas of brains that are thicker, but then we went on to say whether there might be some difference in the way these regions were connected structurally to each other. So we went on to do a diffusion tensor imaging project looking at comparing tone-deaf individuals and controls. And this is just one stereotypical non-tone-deaf subject we're looking at here. So the front, it's like this three-dimensional view like this. So the front of the head is here, the back is here, and left is here, and right is down. And so in the different colors, I've colored the different branches of the arcuate fasciculus, which is a white matter pathway that connects the superior temporal gyrus and the inferior frontal gyrus. Right? So the previous paper found that the STG, the superior temporal gyrus, and the inferior frontal gyrus, IFG, are different in musics. So we went on to ask whether the connectivity between these regions might be different as well. So the different branches that I'm coloring on this brain correspond to the different branches of the arcuate and so what we're seeing here when comparing the control subject against the tone-deaf subject is that the tone-deaf person has much less volume overall, right? So especially in the right hemisphere, there's this superior branch of the right arcuate fasciculus, which connects the superior temporal gyrus and the inferior frontal gyrus in the tone-deaf subject. Well, in the control subjects was not identified in 10 out of 10 of the tone-deaf subjects that we looked at the time. So that was a really pretty striking finding, and it seemed like this has some implications for how the brain talks within itself in people who are tone deaf. Right? After all, these people are relatively normal people. Like they're walking around. They're not kind of being unusual. But this is a pretty striking difference.
So those that are tone deaf, they're lacking the
Yeah, the arcuate fasciculus. Yes, they are lacking the right superior branch of the arcuate fasciculus. Yeah. So, and to follow up on this, we actually placed one region of interest, right, on the superior temporal gyrus. Right? So, and this is kind of this combination of the region-based hypothesis-driven way of looking at brains, and just a more hypothesis-free way of looking at all the voxels of the brain in general. But what we did was we placed one region of interest in the right superior temporal gyrus, and we asked the computer to find us all the connections from that area to other parts of the brain. And what was striking was that while for most normal or non-tone deaf individuals, most of the tracks went from the superior temporal gyrus to the frontal lobe. Right? So that means that the auditory areas are talking to the frontal lobe areas. For tone deaf people, it turns out that these paths went everywhere but the frontal lobe. Right, so you could see the difference, hopefully, on this slide. While for normal people, most of the tracks went from here to here. For tone-deaf people, most of the tracks went from here to parietal lobe and occipital lobe and other places, just not to the frontal lobe. So this really gives us a more networked view of the brain and of this unusual population, where we're not just seeing that certain regions are different, we're seeing that certain regions are just not as linked, or not as actively talking, or not as structurally connected to certain other regions. And then this is just showing that the volume of this right superior arcuate fasciculus was inversely correlated with the threshold for pitch discrimination ability. So in other words, the people who have a high threshold, which means they're not as good at telling the difference between small differences in pitches, tend to have less volume in this pathway. Any questions? Yeah.
Why do you think it is that there are different regions of EEG again in any of those difference in not going to the
Yeah, I think that's a really great question, and people are still trying to find that out. One possibility is that you started different, right? So it might be some kind of genetic difference. Another possibility is that it's a combination of genes and environment. Of course, like you might start out with some vulnerability, and then due to the lack of exposure early on, you don't develop those pathways which otherwise might have normally developed. There's a third view, which is more about neuronal migration, right? So it could be that the right neurons started in the right place for everybody, but due to some early brain event, right? Not necessarily an injury, but just some kind of event. The neurons didn't migrate into the proper synaptic connective patterns. So it might be all of these things, and we're still looking into that. It would be really great to just trace the development of these pathways from infanthood to adulthood, but I don't know of many studies that have really been able to do that. It's very expensive and difficult and so on. And these techniques kind of change over time, right? So, so there was actually a Wesleyan student that worked with me over the summer to collect children's brain images using diffusion tensor imaging to track these arcuate vesicula. And she was able to do it, but we haven't been able to get the next scan and the next scan and so on, and to relate that to behavior. So yeah, so it's an ongoing effort. Any other questions? Yeah. Right. So so there were a couple of papers, right? The first paper was looking at, I think, an ROI analysis comparing different T1 images. The second paper was looking at diffusion tensor imaging, and it was ROI based on a probabilistic tractography. Actually, it was deterministic tractography. So we placed one region of interest in the superior temporal gyrus, another in the middle temporal gyrus, and then said, like, find me the connections between those regions. And the connections showed up as larger in the absolute pitch range. Anything else? Other questions? Okay, great. So I think I'll move on to the chemical senses. Okay, so the chemical senses, when we're talking about chemical senses, I should also note that there's a course on chemical senses that is taught by Professor Helen Treloar, which is very highly rated. So it should, if you find all of this stuff interesting, definitely feel free to take that course in a future semester. But when we're talking about chemical senses, right, so far we've talked about vision, and we've talked about audition, and it's quite clear what the kind of currency is that the visual system and the auditory systems use to communicate information, right? So vision uses light, audition uses sound, or it uses specifically frequency of sounds. Now, when we're talking about the chemical senses, and that includes smell, right, and taste, and also a trigeminal system, which we'll expand on in a bit. But these are the currency that the system uses, right? The physical stimulus that it uses is in response to chemical changes or chemical stimuli. Right? So it's a little bit less well defined, but what we certainly know is that changes in chemical properties of stimuli will result in changes of your perception of these chemosensations. Right? So there are three major chemical sensations that the textbook talks about. The first is trigeminal system, which you may or may not be, you probably aren't as familiar with, but once you hear about what it actually means, you'll be familiar with them. And then the smell, the olfactory system, which is your sense of smell, is also tightly coupled with the pheromone or chemical signaling system, which I find to be very, very interesting. And then there's also the gustatory system, which is responsible for taste. So let's talk a little bit about the trigeminal system. So why do we call it the trigeminal system? It comes out of the cranial nerve, the fifth cranial nerve, which is the trigeminal nerve. Why is it called the trigeminal nerve? Because it goes up from this cranial nerve all the way into the face, and where it trifurcates, is that a word? Where it splits into three. It splits into three different branches. So that's what the tri part of the trigeminal system comes from. And this trigeminal nerve has different functions, right? And that includes more sensory functions, and that's specifically what we're talking about when we're talking about the trigeminal sense. It is especially a way of detecting noxious stimuli, right? So stimuli that are aversive, such as pain and burning, and also kind of a temperature, the sensation of temperature changes to the face. The trigeminal system also is useful for proprioceptive impulses, which is a motor portion of trigeminal nerve function. but just a little bit of a sense of what you're feeling when you're feeling your trigeminal nerve sensations. Wasabi, or the green stuff that goes into your sushi, you know, probably all have this experience. If you eat too much of the wasabi, it feels like it's something that's going up your nose. Right? That is your trigeminal system at work. Okay? Chopping onions, right? How many of you have cried when you're chopping onions? Right? It's like absolutely not really an emotional thing, but just suddenly this sensation that goes into your eyes, and you just can't help but tear up. That is, again, your trigeminal sensation trigeminal nerve sensations at work. So in a sense, that's your trigeminal nerve saying, you know, this is a noxious stimulus, right? This is something that is not good for me, so I should get out of this situation. And even though it might not actually be, you know, a poisonous situation. So that's a trigeminal nerve sensation. I wanted to spend a good amount of time talking about the olfactory system because, you know, it's kind of maybe the next most well-studied sensation. And so the olfactory system, this is a general kind of outline of the anatomical pathway. So of course we use our noses to smell, right? So that means that when there's something that emits chemical sensations, chemical stimulus, that, you know, goes, the air molecules go through the air and get inhaled into your nasal cavity. Right? And so once the air goes into your nasal cavity, you have a few bony ridges that are within your nasal cavity, and the point of these bony ridges is to they're called turbinates. They're responsible for creating disturbances or turbulences in airflow. Right? So that means that when air goes through your nostrils, it gets messed around, or it gets turbinated, or disturbances in airflow happen, and that causes the different chemicals to bind through your olfactory epithelium, which is the skin that's at the top of your nasal cavity, and then through your cribriform plate, and then eventually it gets into the olfactory bulb. And so, just to give you a little bit better of a view of what that means, right? So when we're talking about the olfactory epithelium, that's just the skin that's at the top of the nasal cavity, and that has a set of terminations, terminals from the olfactory receptor cells. And so these are actual neurons that are receiving, that the chemicals are binding onto, and then these olfactory receptor cells then go through the cribriform plate, which is this bony plate that separates the top of your nasal cavity from your cranial nerve and the ventral view of the brain. So this cribriform plate then, so these olfactory receptor cells then terminate, you know, through the axons that go through the cribriform plate towards the glomeruli, which are just clusters of neuronal endings that are inside the olfactory bulb itself. Right? So the olfactory bulb is the first cranial nerve. Right? So if you look at the brain, right, looking at the ventral surface of the brain, the first cranial nerve that you see is the olfactory nerve. Right? And so it terminates in this olfactory bulb, which then talks through the glomeruli and through the cribriform plate into the olfactory epithelium. And so what happens with this cribriform plate is that it's actually quite a fragile part of the human head. And so if you, for instance, don't wear helmets when you're biking, right? Let's say if you fall off the bike without helmets on, you might actually shear, you might actually induce shear forces that cause the cribriform plate to shear through the axons of the olfactory receptor cells, and that leads to the condition known as anosmia. So I've already said that we're going to hear about ablankia for a bunch of times in this course. Anosmia is the inability to smell. So again, the first cranial nerve is the olfactory nerve, right? So that's just at the top of the set of cranial nerves. But a ventral view of the olfactory system here again shows you the olfactory bulb and how it has these terminals that then go through the cribriform plate into into the olfactory epithelium. And what's interesting from the psychophysical perspective is that the air flowing through the different nostrils that then gets moved around by the turbinates, that actually goes in at slightly different rates between the two nostrils. And so that can be measured using a flow olfactometer, which is just a set of laboratory equipment that can assess how airflow goes through your two nostrils at once. And what's really interesting is that while most people have a dominant hand or a dominant foot, right? Most people are right-handed or left-footed or whatever, most people also have a dominant nostril, in the sense that the airflow tends to be faster in one nostril than in the other. And so why is that useful? Well, that's useful for a bunch of different reasons. Right? One reason is if I put a pizza here, as opposed to a pizza right in front of you, you want to be able to tell just by smelling alone how far away that pizza is. Right? And so by comparing your airflow, right, between your dominant nostril and your less dominant nostril, that gives you a sense of how far away something is, right, and how concentrated an odorant is. It also gives you a sense of how viscous something is, right? Viscosity and absorption are inversely related, right? So if something is very viscous, right, you tend to absorb it less compared to if it's not as viscous. So this difference in perception through your different nostrils can give you a bunch of different information, right? Specifically about odorant density and distance and viscosity. So going back to this olfactory system and this problem with anosmia, right? If you have shear injuries through the cribriform plate, that could shear the axons of your olfactory receptor cells, and that could result in anosmia. And so one really interesting paper comes from looking at functional MRI of people, normal people and person with anosmia. And so for this paper, what the researchers did was they acquired scans using this kind of specifically designed tilted scan or diagonal scan, so that they're especially able to localize the activity in the secondary secondary olfactory cortex, which is the lateral orbitofrontal cortex. And so the primary olfactory cortex is the piriform cortex, which is right next to the olfactory bulb. And then beyond that, what we tend to get those regions projecting to the lateral and orbitofrontal cortex. So what these researchers found was when they were giving odorants, right, so things like chocolate or coffee or whatever, to their participants and asking them to sniff. And sometimes there were some control conditions where there was no odorant, it was just clean air, and people were asked to sniff. It turns out that the primary olfactory cortex, which is the piriform cortex, was active regardless of whether some odorants were present. So whenever you were just sniffing, you tend to activate your primary olfactory cortex. However, when you were actually smelling something, and that there's actually some odorant present, you activate the secondary olfactory cortex, which is in the lateral and orbitofrontal cortices. So that's pretty useful, right? So why would you want a system that is just detecting sniffing? Well, again, this goes back to the idea of the near pizza versus the far pizza. Right? If you have a gigantic pizza that's very far away versus a small pizza that's very close, how hard you have to smell to detect the same level of odorant needs to be coded for, right? So that piriform cortex, the primary olfactory cortex, is actually doing that kind of weighing. So what about a person who has anosmia? Right? So they also got fMRI data from one subject who has unilateral anosmia. This is really unusual, but this is the case of just lacking the sense of smell from one nostril, right? Which could happen as a result of certain kinds of injuries. And what they saw was bilateral activation in the piriform cortex, which again is the primary olfactory cortex, when this person was sniffing through their good nostril, as opposed to no piriform activation when they were sniffing through the anosmic nostril. So this tells us that the primary olfactory cortex is bilateral, even though your input might be unilateral. Cool. So so this leads us to another kind of closely related but different topic, which is how do we use our senses? How do we use our chemical sensation to signal to ourselves and to each other, right? So this is the topic of pheromones, which is chemical signaling. And we know that in the animal kingdom, we use pheromones as a territorial or mating behaviors, right? So we're probably familiar with dogs that breed to mark their territory and so on. But the real question for cognitive neuroscience is to what extent this also exists in humans. And so it turns out that there's a bunch of different evidence for possible existence of pheromones, a pheromone system in humans. So women to women, there's plenty of evidence that menstrual cycles tend to synchronize in dorm rooms, right? So a bunch of women are living together in a dorm, then their menstrual cycles tend to synchronize. And there was this classic study by Martha McClintock, who was at Wellesley College. What she did was she got I think it was sweat from t-shirts of one group of women, and then rubbed them under the nose of another group of women, and then lo and behold, the second group of women who sniffed the pheromones of the first group of women, their cycles synchronized with the first group of women. Right? So this is an example of women to women pheromone signaling. But then there's also plenty of evidence for male to female and female to male pheromone signaling. Yeah, go ahead.
How many, like during that experiment, how many times did they have to sniff the thing before they cycled?
Oh, that's a really good question. I don't remember that off the top of my head. But why don't I post that paper onto Moodle and then and you can look at it. But yeah, but I actually don't remember. It's from quite a few years ago. But we know that it's a pretty strong effect. So, but you're right that it might take a little while. Yeah. This next one we know to be a fairly just a one trial kind of thing. But there's most women will say that they can recognize their male partners through smell. So this is an example of male to female chemical signaling through pheromones. And most women will also say that they can pick out close family members via smell. The opposite doesn't seem to be true, and we're still trying to figure out why that is. This one is pretty crazy. This is a female to male signaling. So exotic dancers, the tip money they get from exotic dancing actually correlates with their menstrual cycle. So when women are ovulating, they're actually a little bit more likely to get more tip money. Don't ask me how we found this out, but that is one example that has been cited as one example of women to men signaling. Now, of course, that can be quite a confounded situation. Maybe they're just dancing more confidently. I don't know, but this is another one of those where you can't really tell. And it is that men with anosmia have fewer sexual partners. Right? So if you're worse at detecting these pheromones or these chemical signals, you're worse at mating. Now, this last one I'm going to go a little bit into because it's a fairly recent paper that I think is just really kind of mind-numbing. There's some new evidence that human tears could actually contain a chemical signal. Right? So when we're talking about pheromones, most of the time you're thinking about like sweat or you're thinking about other kinds of bodily functions. But tears, this one group thinks that they actually contain an emotional pheromonal signal. It's actually a chemical signal through which people can communicate with each other. So how did they find that out? One of my favorite studies, really quite mind-numbing. Okay, so these researchers, I think they're in Israel, they got a bunch of women to come in and watch sad movies. Okay, so how many people cry when they watch sad movies? Okay, great. Right. So then they collected these women's tears. So I don't know if it's because they're in Israel or what, but they only got enough women to cry to sad movies. They couldn't find enough men to cry to sad movies. They kind of stereotypical stuff. But anyway, so they got these women to cry to sad movies, and then they collected their tears, right? And then they also collected saline solution, which had been dripped over these same women's faces, right, just as a control. So not tears, but similarly salty fluid. And then they made the men, a group of male subjects, sniff these little cotton pads that were either drenched in women's tears or drenched in the saline control solution. Okay? And then they made people do these pictures rating tests where they were given these pictures of women's faces and asked things like, to what extent is this face sad, and to what extent is this face sexually arousing? And amazingly, men who had sniffed tears rated these faces as more sad and less sexually arousing compared to their saline matched controls. So that's pretty amazing. And what's even more amazing is that they then did functional MRI of these male subjects and while they were watching pornography, right? So they're actually watching erotica, and it turns out that men who had sniffed the tears as opposed to the saline control showed less activity in the hypothalamus, which is from one of the first days when we're talking about dissection and different cell sizes in gay versus straight men. We know that this hypothalamus is linked to sexual arousal. Right, so there was this reduced activity in the hypothalamus while these men were watching a lot of films. So it's pretty amazing finding. So the New York Times, then of course, when this paper came out in Science, they did a little write-up on it, and their punchline was, tears are a woman's way of saying, not tonight, dear. So you use tears. Why do women cry? Because you're using your tears to convince men that you're not interested, you're not sexually interested, you are just sad. And so men are using that signal to kind of understand that there's more sadness and less sexual arousal that needs to happen tonight. Pretty amazing. Any questions about that? Okay. All right. So let's move on to talking about the gustatory system. So obviously, these are major topics of research, and in a sort of survey course like this one, I try to cover the general stuff, and then I try to pick out a couple of super interesting papers to show you guys. But of course, if you're especially interested in one of these topics, feel free to look into the textbook, and you'll find some more references, or you can come talk to me and find some more references, or you can take an advanced course on the topic. So the taste system and the gustatory system. So of course we know that we're starting with taste apparatus, and for humans, the taste apparatus are on the tongue, and that they're a set of papilla or papillae that are actually tiny protrusions on the surface of the tongue, right? And so when you zoom in on one of these papillae, you can see a set of taste buds, which are the taste receptor cells, which are lining the sides of these papilla. And these taste buds are distributed for different flavors and different tastes are distributed differently across the surface of the tongue. So we know that sweet sensors, for instance, are more distributed towards the front of the tongue, and sour sensors more distributed to the sides of the tongue, and so on. There's a fifth taste that we know that from multidimensional scaling studies, which are psychophysical studies, we know that is not completely captured by the combination of these four other, and we just call it umami, not otherwise well defined. But in all cases, there's a few cues that we use to identify a flavor, right? And that's flavor identity itself, and concentration, and also hedonics. Right? So we can quite quickly determine whether whether a flavor is something that we like or something that we don't like, and we use that to associate with the identity of the item itself. So at the neural level, what's the anatomy of this whole system? So there are a couple of cranial nerves that go into the tongue. One is an ossicrandial nerve, which is cranial nerve 9, and the other is cranial nerve 10, which is the vagus nerve. So it's important for facial movement as well as tongue movement. And so those are also part of the taste system. And so then from these cranial nerves, they go up the solitary tract, which is a major projection fiber that goes up your brainstem and midbrain towards the thalamus. Okay. And then at the level of the thalamus, we have axons that then project to the gustatory cortex in the primary gustatory area, which is in the frontal operculum and the insula. Right. So the insula is one of these giant areas that are kind of throughout the side of the brain. Right? So if you're looking at the side of the brain, if you kind of pry open the temporal lobe from the rest of the brain, you expose the insula. And so the anterior part, right, the part that's closer to the front of the insula, kind of goes towards the frontal operculum, and that area is known as the primary gustatory area. So what do we need in a taste lab? So there's actually a Wesleyan grad who spent a couple of years doing this MRI of the gustatory system. And so it's a really interesting setup, but you go into an MRI and you actually can, it's called the milkshake task. You get a bunch of little syringes that are designed to deliver milkshakes of different flavors in very measured quantities. And so it's measuring. So if you're, for instance, interested in why people are obese, right? You might have some people who are obese going into the scanner and tasting different milkshakes and looking at their reward responses to it and so on. And then there are more sort of larger apparatus, such as this BOD-POD, which is for measuring BMI or body fat. And then there's good old behavioral experiments, and I really like this picture because it's showing you how eating can be thought of as not just a behavior, but a social activity and an emotional activity, right? Because there's so much emotion and social referencing going on there. So it's not just eating as a kind of regulatory behavior, but it's a kind of, the relationship that we have with food is a much more social and emotional thing in humans. And so you could ask this question of, when you're stealing food, does it taste as good, or does it taste better? Right? Or when does food actually stop tasting good? Right? So when does food actually, how does food actually interact with the emotion and reward systems that we all have? So this is really a fun study that came out quite a few years ago now. And this is one of the very few PET studies that we're going to talk about in this course, so positron emission tomography. And so the time resolution, of course, is not great because the PET studies are limited by the time resolution that the technology has. But what they did is really kind of brilliant paradigm. They found chocoholics. So these are people who love chocolate. And so they wrote and they got people who say on the scale of 1 to 10, I'm an 8 for loving chocolate. 8 and above, right? And then they gave them chocolate, and they kept feeding them. So they started with just a little bit of tasting water, and then over a long period of several minutes, right, they could eat as much chocolate as they wanted. Right? So that's the CHOC1 portion, right? And so they would eat and eat and eat. They would feed themselves as much as they wanted, and then until they're done, and then they would rest. And then the second block, they would eat again as much as they wanted, and then they would rest. And the third, they would feed again as much as they wanted until they got to rest, until they're done, and then they would rest, and so on. And so this happened 7 times. And what happened after each of these feeding periods was they had to rate on the rating scale how much they want to eat first, how pleasant they found the chocolate to be, and how much they would want another piece, how motivated they were to keep eating. And not surprisingly, they started out, loving it, great chocolate, really, really good, pleasant. I would love to eat more, and then it decreased. But what was fun was that there's this divergence in the decreasing rating functions. And it turns out that after 7 feeds, people were still saying, "Yeah, it is still very good chocolate. I'm just absolutely disgusted and just don't want it anymore." Right? And so this is really you can really map out the time course of this conversion from pleasure to aversion of eating chocolate. And what they found was, the specific regions aren't so important for this as much as it's the trend of decreasing activity over the 7 chocolate feeding periods. What they found was that the orbitofrontal cortex in the frontal lobe, which is important for motivation and reward, and also the ventral tegmental area, as well as the primary gustatory area, they tended to decrease over time, right? So as the subjects went from being pleasured to being averse to chocolate throughout the course of the study, they also saw decreasing functions of these reward and taste-related areas. All right, so let's see. So this goes till 2:40. Okay, so we got a couple. I think we got a little bit over 20 more minutes, and I think we'll cover the last part, which is mechanosensation. So in addition to chemical senses, we also wanted to talk about mechanical senses today. So mechanical sensation is the mechanical stimuli acting upon the body. And so we know that the skin is the largest organ that we have. And within the skin, we have a number of different sensors. We have a number of different receptors, multiple receptor types that are sensitive to different kinds of mechanosensation. And so that includes the cutaneous or subcutaneous system, right? So that's sensitive for touch and vibration and pressure and tension. And then there's the nociceptive system, which we already talked about a little bit when we talked about trigeminal senses, but that's important for sensing pain and temperature. And then slightly different ones are the proprioceptive system, which is important for sensing position and for body movement. So if you're a dancer, for instance, you might have a very strong proprioceptive system because you need to know which part of your body is where at a given time, right? Then there's also the vestibular system, which is especially coded for in the semicircular canals in your ear, right? And that so that's quite close to the cochlea, which we talked about last time, in your inner ear. And what that is useful for is sensing your head position, right, and your acceleration and position of the head, and also your balance. So you don't have to memorize all of these different names of the nerve endings, but we just need to know that there are multiple receptor types that are differently distributed throughout the body. Right, so we know that, for instance, your 2-point discrimination threshold on your fingertip is much smaller than your 2-point discrimination threshold on your back. Right, so if I'm touching you with 2 points, those can be a lot closer in your fingertip for you to be able to tell that there are 2 things, as opposed to if you're doing the same on your back, right? And so these are differently distributed throughout the body, but in all cases, the primary somatosensory pathway, it goes up, it's differently coded for in your upper part of your body, for your head and neck, as opposed to the lower part of the body. And again, this is just out of your textbook. There's a number of different way stations in the spinal cord. But by the time you get to the brain, what's really important here is that there's the thalamus, which has the ventral posterior nucleus, which includes the lateral portion, which is important for sending signals from the lower body. and the medial portion, which is important for sending signals from the head and neck. Right? So they're differently coded at the level of the thalamus. But then at the level of the brain, you have, this is the super important part, you have the primary somatosensory cortex, which is important, which has these different components that are sensitive to cutaneous and tactile proprioceptive senses. But then those get integrated at the secondary somatosensory cortex, which is S2, and so that's the most important area for sensory, for integrating sensory information from S1. And so the other part of this is something we've already talked about back in the first day, I think. So this is from the Walter Penfield paper of results on different motor homunculi. You can also have somatosensory homunculi. And so that just means that there are different parts of the brain, different areas of the brain that are coding for different sensations from different parts of the body that are proportional to their sensitivity. Right? So areas of the body that are more sensitive have larger neural real estate and are dedicated to that. And so the homunculus, the somatosensory homunculus, is just a representation of the body where the size of that each part of the body is proportional to how much neural real estate is devoted to it. So lips, for instance, there's a lot of space in the brain that's devoted to sensation on the lips, and so that's a large representation, and so on. And so this kind of relationship between the brain real estate and different part of your body, that's known as somatotopy. Right? So we've talked about retinotopic mapping when we talked about vision. Hope we talked about retinotopic mapping when we talked about vision, which is the systematic relationship between space and the part of your visual cortex. Right? And then in audition, we talked about tonotopy, which is the systematic relationship between frequency and your auditory pathway, or a certain part of your auditory basilar membrane. But in the somatosensory cortex, you can talk about somatotopy, which is the systematic relationship between your body and brain parts that are devoted to processing sensations from that part of the body. And so that leads us to playing with certain tricks that kind of make use of this somatotopic, this idea of somatotopy, which is the relationship between somatic sensation and brain areas that are responsible for detecting these sensations. So can I get one volunteer? Okay, you can come up. What's your name? All right, so you can sit right here. And yes, so I wanted you to sit up here before I was going to. Okay, so are you left-handed or right-handed? Okay, let's use your left hand. Go like this. Okay. Okay, so I want you to. Okay, let me make sure I can do this. Okay. Okay, can you close your eyes? I'm going to touch your arm very quickly, and then I want you to open your eyes and tell me where I touched you. Okay? Ready? Okay. Tell me where did you get touched? You can open your eyes.
Several places.
Yeah. Okay. So. Okay. Okay. One more time.
Yes.
Okay. Okay, great, thank you. That's all. Thank you very much. That was a very brave volunteer. Yes, if you have been, yes, I like to do these kinds of activities throughout. So, yeah, this turned out to be the first one of this semester. So this is called the cutaneous rabbit illusion, and feel free to try this out with your friends. And so the idea is that you have different parts of your brain that are next to each other that should be responsible for sensation all along your arm, right? And so I did 2 trials, right? And in the first trial, I did 2 touches here, 2 touches there, 2 touches there, which is the control condition, right, where you get these 3 positions and you get these 3 touches that are along the surface of the arm. Now, in the second trial, I did 2 touches there, and I did 4 touches there, and then 2 touches there. So that's more like the illusory rabbit condition, as opposed to the vertical rabbit condition, right? So a good demonstration of somatotopy, which is the cutaneous rabbit illusion, a person should feel as if they were getting sensation to touch here and then here and then here. Right? So it's, even though you were only being touched at the beginning and the end of the arm, you should feel an illusory touch in the middle of the arm. So that seemed to have kind of happened. It seemed to have worked out quite well. And so feel free to try this. And what this group showed using functional MRI was that at the moments when people were supposedly feeling the illusory sensation, the brain regions that were responsible for the second vertical sensation in V2, which is the middle of the arm, were actually more active. Right? Again, suggesting that they were actually feeling this this illusory sensation in their somatotopic, somatosensory cortex. Cool, right? Okay, so the last thing I want to talk about is the pain pathway. So we already know that there's the general anatomy of this pain pathway is not really so different from the somatosensory pathway, where it goes through different parts of the spinal cord towards the thalamus, which then goes through the ventral posterior medial and ventral posterior lateral nuclei of the thalamus towards the brain. But once you get to the brain level, there's a lot of very interesting work on how we feel physical pain, right? What parts of the brain are the neural signatures for physical pain. And so this is done using functional MRI while people were experiencing mild to moderate forms of pain through blood pressure and so on. But in general, in addition to the primary and secondary somatosensory cortices, which is S1 and S2, we also have a few areas that tend to get active at the same time. And one is the anterior insula. So that's right there. And another is the anterior cingulate cortex. So the cingulate cortex generally is right above your corpus callosum. So this is your corpus callosum. On top of that is the cingulate cortex. And the anterior cingulate cortex tends to be active along with the anterior insula and the somatosensory cortices when people are experiencing physical pain. So that's the fMRI-based signature of physical pain. But what about watching other people in pain? Right? So this is a really interesting, I think very interesting, fascinating area of neuroscience research that we'll kind of revisit when we talk about social cognition. But it turns out that when we're watching somebody else experiencing pain, we get some of the same regions being activated as well. And so specifically not the primary and secondary somatosensory cortices, but the anterior insula and the cingulate cortex also tend to be activated when you're watching somebody else experience pain. And so that kind of gets people to kind of philosophize that watching somebody else being in pain is as if you're feeling pain yourself, right? But it turns out that that relationship is quite an interesting one, and it's moderated by how close you actually feel to the other person. So like watching a person I don't like feeling in pain is quite different from in the brain from watching somebody I do like feeling pain, which is quite interesting. And so we'll talk more about that later in the semester. But I do want to say a couple of things about brain plasticity and what we can do to either alleviate someone's pain or to rewire the brain so that abnormalities kind of get reorganized. And so this is a really classic study from a very classic study on owl monkeys where they took the somatosensory cortex and then they amputated a digit of a finger, right, of the hand, And so what the researchers saw was that the corresponding part of the somatosensory cortex after amputation got rewired such that there was no longer a neural representation of the amputated digit. Right? So while the middle finger originally had its representation in the somatosensory cortex, sometime after being amputated, the neighboring regions actually took over and for a region that was no longer there. So, and that has very kind of dramatic implications for people who had been amputated, due to being in the front lines in a war or some other reason, right, or some accident. So this is a very interesting example of an amputee who would actually report feeling sensation in a part of the body that's not there when they were touched in a different part of the body, right? So this is a person who no longer had a left arm, right, due to amputation, and they would say that when you touch their face, they would actually feel illusory sensation in that missing part of the body. So, in contrast, you can also have an expansion of a somatosensory map during, as a result of extensive use, right? So one example is pianists, right? For instance, because they play piano a lot, and so that uses their hands and fingers a lot. You end up having larger neural representation for those digits, right? For the fingers that you use. And so if you compare people who use the left hand to play music versus right hand to play music, right? They show different patterns in the somatosensory and motor cortices. But so I had a very good example, actually, I'm very sad, but good example of these kinds of rewiring situations is phantom pain. Right? So after people, for instance, amputees are coming back from the front line, who might be missing certain parts of their body, sometimes still feel a phantom pain in a part of the body that's no longer there. So these are some examples of amputated parts of the body, and then the colored parts are just where people report sensations. I actually have a very good friend of mine had his middle finger amputated because of a lawn mowing accident. So it was a friend of mine from college, and when he was 12, he was driving a lawnmower, and he's from Texas, where I guess they own a ranch in Texas, and he was driving this lawnmower, which you shouldn't let a 12-year-old drive a lawnmower, but anyway, so he was, and then and he saw that the lawnmower got stuck, and so he reached his hands into the lawnmower, and it suddenly got unstuck, and then he looked at the back of his hand, and there were these little scars on his middle finger, and then he looked at the front of his hand, and he could see the bone. And so he got rushed to the hospital, and after a long time, they decided that the best thing they could do was amputate that finger. And there was a very long story of like having it sewn to his belly so that the skin from the belly would actually graft onto the hand, and eventually he just had a scar there, but like just like this, right? Otherwise he's a normal person. He was left-handed writer anyway, but so he's like this, and otherwise totally fine. But we would be driving together, and like sometimes he would be driving, and he would like be rubbing on this finger that's not there, and I would be like, "Why are you doing that?" And he would say, "Well, it's because I'm feeling sore. I feel soreness and pain in that former finger." And it's like, "Oh, that's just what I learned in cognitive neuroscience." And it turns out that this phantom pain is actually quite common. So neuralgia, which is just a word for pain, can happen in amputated limbs. So what can you do to help that? Ramachandran, who's a neuroscientist in UC San Diego, famously created this very clever idea of just creating an illusion, right? Which is providing a visual feedback to enable multisensory integration, so that you're basically tricking the visual system to think that there is a part of your body that's there, even though it's not actually there. So you're tricking yourself into thinking that you have a certain part of your body even though you don't. And so that's the mirror box therapy. And so this is a person undergoing mirror box therapy. This is an actual person undergoing mirror box therapy. It doesn't always work, right? But the idea is that you put a mirror there so that your mobile limb then looks like your missing limb. And so by giving yourself visual feedback of as if you had a missing limb, you would actually be able to flex a good limb, which makes you feel like you're flexing the missing limb, which alleviates any pain or soreness that you might have been feeling all along. So it doesn't always work, but when it does, it's really great, right? Because mirror is obviously a very cheap